This is the colorful slide that George Daly was talking about. And it is a thank you slide as much as a conflict of interest slide. And thank you, all of you, and everybody that helps turn our sometimes half-baked or crazy ideas into things that actually have an impact in society. And we've been fortunate that many of the things we work on turn out to be exponential technologies. And what that means is it can change by 2 to 10-fold per year. So they're going exponentially improving in quality and price faster than electronics, which is already a mind-boggling rate of change. And this includes reading, writing, and editing of DNA, 10 million-fold change in price, and somehow people still manage to make money on it even after it's brought the price down 10 million-fold. And so we brought the $3 billion genome, which everybody talks about as Genome Project, I'm embarrassed by. I was not a big, I started it in 1984, but I wanted to bring the price down. It did eventually come down to less than $1,000 about 4 years ago, and Veritas Genomics was one of the companies that provided genetic counseling. But now we think we've got a business model where we can give away genomes for $0. And this, like many things I'll mention today, may seem science fiction, but it is. Many of these things are past or present tense. And you think of this like maps and search on the internet. Those are free to the consumer. Somebody has to pay for it. But with genetics, there are a few ways that we've discovered that I think are plausible. So these are just technically, these were all done not by filling a room full of machines or technicians, but by reducing it so that one tube, one pipette, whatever you have, now does billions of reactions with the same effort of doing one. So it doesn't, you can scale that up if you want to the size of a room, but the important thing is we scale it down to billions of reactions in a single small tube. Now the themes that I'd just like to develop here, arguments in a certain sense, is there's an immediately accessible revolution in genetics we don't have to wait for, which is monogenic or Mendelian diseases. These are so-called simple, nothing simple, but these can be done by orphan drugs, by gene therapy, and I constantly encourage us to think about genetic counseling as a cost-effective alternative to gene therapy, even though a huge fraction of my research is on gene therapy. But you can bring down the cost in ways we'll talk about in a moment. Then there's the alternative, which is multigenic, multifactorial, environmental. And these are sometimes called common or complex or chronic diseases. And I think many of these should be or are being rebranded as diseases of aging. 90% of us in industrialized nations will die of these kind of diseases. But it doesn't necessarily, just because it's a complex disease, doesn't necessarily mean it's a complex treatment. And I'll give an example of that. And then there's the question of whether we're doing prevention, whether we're doing reversal, partial reversal, or full-time cure. We're mostly focusing on reversal, as you'll see in a moment. And whether this is disease-specific, or we're taking one aging disease at a time, or whether we're coming up with a general set of aging-specific. And we can do this with cell therapies or gene therapies. Now here's an example of the textbook classic multigenic trait, multi-environmental trait. Tens of thousands of single nucleotide polymorphisms, and it seems hopelessly complex both diagnostically and therapeutically, but there are some genes that have very large effects, and these can be turned into therapies, most notably human growth hormone, somatotropin, which is a single gene product protein that works on all these different diseases used clinically. So you might think this is a silly question for me to be asking. Is human genomics useful today? But I will ask for audience participation. How many people here have their full human genome sequence available to them today? That is a truly remarkably high number and disappointingly low number. It's typically less than 1% in the audience, but I will argue it is useful. But it's useful in a curious way that we'll get to. The cost for one of these Mendelian diseases, and there are about 7,000 of them, has been estimated somewhere, depending on whether it's orphan drugs, it can be $100,000 to $1 million per year per patient. So a lifetime bill might be as high as $20 million. And the numbers vary. I give a citation here from Canadian reference. But the point is it's high. And this can be recovered if we can avoid Mendelian diseases through genetic counseling or other means. This can be preconception. It can be even premarital. And the reason I'll say that this is quirky is I think it's analogous to seatbelts. Is we had seatbelts were essentially free. They were standard equipment. They were required by law. There was ad campaigns. Nevertheless, people did not buckle up until this little piece of technology was added to the seatbelt, which the seatbelt wasn't just a passive lock. It actually triggered a circuit and stopped an annoying sound. So we need the equivalent of an annoying sound for human genetics, which, like seatbelts, only 1% of people will extremely benefit, but that 1% is such a big deal that they should do it, and you don't know that you're going to be safe, that you won't be one of the 1 million people a year that dies of car accidents or Mendelian diseases. Now, when we do gene therapy, we have options of addition, subtraction, precise editing, and epigenetic, meaning regulatory. There's been a lot of attention recently to CRISPR, which is mostly subtractive, but I think we need to think about the whole gamut of possible interventions. And for aging, we've heard a beautiful description of and show some of the data behind this 9 or 10 different pathways that are critical for longevity and or aging reversal. But we have such deep knowledge of these, so you can look at it half empty, half full, but we have such deep knowledge, I think that we can start turning these into therapies quite easily, and the route that we've taken is gene therapies, you'll see in a moment. And this includes reduction of cells that have genomic damage or are senescent, the telomeres, the protein regulation, caloric restriction. You've heard of many of these already. So I'm going to go through very, very quickly. I just want you to see 8 or 9 examples here, and look for the word reversal in here, because I want to say that this is past tense. We have examples already shown in animal models, and now we want to convert these into therapies that are suitable, safe, and effective for humans. So here's a couple of examples of small molecules, rapamycin, metformin. I think Nir will be talking about this later on. Here's an example where there's 4 transcription factors, more about transcription factors in a moment, that regulate processes, and these have now been turned from something used in cell biology in the tissue culture plates into an experiment that has been done in mice, where you get amelioration of many age-associated hallmarks. Telomerase can do reversal, and in particular cases, if you have cancer-resistant mice messing around with tumor suppressor genes, you can also have telomerase have its impact without much risk. We've heard about senolytic methodology, the P16 pathway. There are actually the dark matter of the genome, the repetitive elements have been implicated in this sort of senescence. And we have a lot of work on almost every major category of repetitive elements. These are the parts that most genome people don't want you to talk about. It's like the bad secret. Limb regeneration happens in some animals, and there is efforts to see if this can be translated into various regenerative methods in humans. There is this heterochronic parabiosis, which is jargon for fusing the circulatory systems of 2 animals, or just taking the blood itself. And this has been shown to be valuable for reversing cardiac and skeletal muscle, neuronal function, and bone. Another senolytic. Every time you can count the number of atoms, that means it's a small molecule, but our emphasis is on larger molecules. This is work from David Sinclair, with whom we collaborate, and it's the segue into the first of our large molecules. This is the mitochondrial pathway, which George Daly mentioned, the Haggis group is working on, as well as David's. And this is nicotinamide is one way of restoring the inevitable decline in a number of small and large molecules that drop between age 6 and 22 months in mice and corresponding aging in humans. So we tackled one step in this, the TFAM down at the bottom there, which results in loss of mitochondrial homeostasis, and we decided we would tackle it directly. Now using CRISPR not in its normal role, which is hatchet man, where it just kills genes, but in this case we knocked out the nuclease and attached it to a regulatory domain, so it can now imitate transcriptional activator regulators and targeted for 4 different sites upstream for the TFAM, which itself is a transcription factor regulator. Sorry if there's too many levels of regulation here, but it's all good because we managed to restore, there's a 2-fold decrease in mitochondrial function via this pathway. We restored this up to 8-fold, which is more than we need, and so we have some working room for modulating this. One of the advantages I should mention, that one of the reasons we keep getting attracted to gene therapy is that it is something that, unlike small molecules or even protein therapies, the orphan drugs, this is something you can do once in your lifetime and it can have a lasting effect. It's something that you can target very specifically to specific cells or even within a cell, and you can have feedback systems. It's a much more sophisticated drug, and I think of it as like the difference between a simple transistor and a modern supercomputer. So what we've done to generalize that a bit, and here we're restoring protein functions as they decline with age for the most part, just by boosting them up. And so we looked through all of the genes that have these sorts of properties, that have experiments that have been done in a variety of animals that indicate that they are, there's evidence for either longevity extension, sometimes up to 2-fold or more of the lifespan, or aging reversal in various animals. And they're systematically turning those into gene therapies, which is remarkably easy to get a proof of concept. Noah Davidson, pictured here as a postdoctoral fellow, was almost single-handedly did 45 gene therapies, and then using them in various combinations to see, because we want to get all of those 9 or 10 pathways of aging, because we think that it's naive to think that just one of those pathways will be sufficient, but it might not be that hard to get 9 of them with a small number of genes. So using 2 or 3 at a time, we have now, Noah has found a combinatorial gene therapy, which is under review at Nature Biotechnology right now, which hits 5 out of 5 of these age-related diseases, which is indicative of, but is not quite proof, that this is getting at some fundamental aging process. So this is high-fat, diet-based obesities. We're sort of inducing accelerated aging in many of these cases: type 2 diabetes, osteoarthritis, cardiac damage model recovery, and kidney damage. Now, part of the reason those gene therapies were restricted to a subset of the genes in Pedro de Magales' gene age database. He was a previous postdoc in my lab. It was restricted to the subset which are so-called non-cell autonomous, meaning that they have actions that spread to adjacent cells or throughout the body. That's a small subset, and it's because adeno-associated virus is not yet fully tamed. It is among the best delivery systems that we have. It is the most approved method of gene therapy delivery, but it has room for improvement. And we have now combined machine learning, artificial intelligence, and synthetic biology loop that allows us to engineer now over 250,000 different viral designs. So these are not random designs. These are machine learning based designs where we've made specific many specific changes to the AAV virus, looking for improved resistance to immune problems and to tissue distribution. So we can inject this mixture of engineered viruses into an animal and then take samples from throughout the body and find which tissues it's going to and develop new viruses. And this is the work of Pierce Ogden, who's a graduate student, Eric Kelsick, who is a postdoc in transition. The hypothesis that we have, one of the hypotheses that we're using in this work is that we can go from almost any cell type and age to almost any other, notably from pluripotent stem cells to any other tissue. And we have examples where you can go from 70, 80-year-old tissue to almost embryonic and the other direction. And the most dramatic case of this is Bruce Yankner's paper on which I was a co-author that just came out a few days ago. George Daly mentioned Bruce's work on a different subject, where we could take people with Alzheimer's disease, late onset and so-called sporadic, and healthy age-matched controls, and establish stem cells for each of them, and show that their RNAs are almost identical, which is a nice control for the next step, where we turn them into neuronal progenitor cells and neuronal networks, and now they're dramatically different. So even though the sporadic means that by definition they don't have the classic genetic markers, we can nevertheless classify these into people that will get Alzheimer's and those that don't. And more important than just the diagnostic component here is that we can use this as for testing new therapies. So here's an example of differentiating human pluripotent stem cells. These actually are from my body. It's more ethical to do experiments on me than for me to do experiments on my students. Anyway, this is Alex and Paris Dudikoskalaga are recipients of Blavatnik support, early adopters in this accelerator, very grateful. And here is producing endothelial cells and bipolar neurons, initially in separate media, and then later combined together. This was actually harder than it looks. All those little red lines are very thin capillaries, 5 microns, just like the capillaries in your body. There's almost an equal number of capillaries and neurons in your brain, little known fact, 86 billion of each, and that's what you're seeing here. Is you have these big nuclei for the neurons and the smaller red capillaries floating through them, the blue nuclei for the neurons. We also have, so that's an intimate connection between two different cell types that we developed from a single cell type. And here's two other ones where the oligodendrocytes wrap the neurons with myelin. You may have heard of this as speeding up the conductances through the nerves that go through the white matter in your brain, so you can go long distances at high speed. This is beautiful wrapping that looks just like natural. So one of the example of a success story for gene therapy is CAR T cells. Hopefully you've heard of this. This is not only a way for T cells to fight a variety of cancers, but it kind of shows off how how genetic editing can help with gene therapy in making them much more immune compatible, so you can actually transplant from any person to any other person, sometimes called allogeneic or universal cells. And we generalize this even further from any person to any animal to any person. And we're in the process of testing this now. We made dozens of changes in the pigs to make them humanized and missing the endogenous retroviruses, so they were less of a risk. And this is now in preclinical trials in non-human primates at MGH and other places using these pigs. And to do this, we had to overcome natural inclination for cells to die when they're cut multiple times with CRISPR. We made so many changes to the cells, we had to develop ways to stabilize them and make them happy with that number of edits. And we've now set the record at 62, and the new record is now at 15,000 edits in a single cell. It's not published yet. You heard it here first. And finally, I just want to say that part of the attraction for engineering organs is not just to deal with the incredible worldwide shortage. where millions of people could benefit. It's the opportunity to do something that's very hard to do in humans, whether they're human organ donors or just humans in a preventative medicine sense, which is to make us resistant to multiple pathogens, to make us cancer resistant, senescence resistant, immunity that we've been talking about, and even cryopreservation if you want to bank away organs. This is very hard to do, but we know how to do it in animals, so what if we could transplant it from animals to humans? And here's some examples of some animals that can survive through full body freezing. So that's all I want to say. Thanks again, not just to the students and postdocs I mentioned along the way, but here's Bruce Yankner, who's helped us with a number of these neurodegenerative diseases, and Jennifer Lewis, who's helped with the organ. So thank you very much, and open it up for questions. Thank you.